FluoRender tutorials. Selecting and extracting structures. Part 1. Paint brushes. You can select structures using FluoRender's paintbrush tools. Click Paintbrush from the main toolbar to open Paintbrush setting window. Select and isolate the green channel from the Workspace panel. Zoom in and find the structures to be selected. We would like to select some of these cells. Click the Select button to enable the selection brush. You can use the sliders under Brush Properties to change the size of the selection brush. You can also use the mouse wheel to change the grow size when the mouse cursor is in the render view. Click and drag the mouse to select the desired structure. The selection brush uses two circles to define two regions, one for seeding and another for diffusion. To fine-tune the selection and include more low-intensity signals, lower the edge strength and enable the diffusion brush. Paint with the diffusion brush and add more to the selection. You can also lower the threshold value to include lower intensity signals. You need to click the brush type button again to allow normal render view interactions. The selected part is rendered with the volume channel's secondary color and composited with the primary color. You can change the secondary color for the selection in the property panel of the current channel. Click the Unselect button and paint in the render view to remove structures from the selection. Use a combination of different brush types for satisfactory selection results. Click Reset All to clear the selection. Enable Auto Thresh to let FluoRender automatically determine the threshold value for selection. You can use a series of keyboard shortcuts for the brush tools, which allows fluid switching between brush tools and view interactions. Hold down the Shift key while painting for the selection brush. The auto thresholding function calculates a new threshold value for each paintbrush action based on the selection's histogram. Only one structure is selected for each paintbrush action. The Edge Detection option enables or disables the edge strength setting, which is used to separate structures. A higher edge strength will be more likely to stop the diffusion at a gap between the structures. When the Visible Only option is enabled, occluded structures will not be selected by the paintbrushes. FluoRender will only set seeds on visible structures and diffuse the seeds based on connectivity. Disable Visible Only and select occluded structures. Both the front and rear cells are selected now. Leave Visible Only on for more intuitive paintbrush selections. Part 2. Deletion and Extraction. You can create new channels by deletion and extraction. Select a cell using the selection brush. Click Erase to create a new channel with the selected cell erased. The original channel is turned off. Turning its display on reveals the selection as well. Click Extract to create a new channel to only include the selection. Go back to the original channel and repeat selection and extraction to segment two more cells. Create a new group for the extracted cells. Change the colors of the new channels to distinguish them. Part 3. Saving extracted channels. Saving the current progress as a project also saves the new channels containing the extracted cells. Click Save Project to save the project. A file folder is created in the project files directory. It contains all the newly created channels from the original data. In FluoRender, clear all loaded datasets. Drag and drop the files in the project folder into FluoRender. The first channel is from the Deletion. The rest channels are the extracted cells. Part 4. Undo and redo paintbrush operations. To use undo and redo of paintbrush operations, you need to enable paintbrush history, which is disabled by default. In the setting window, increase the setting for the paintbrush history. Set the number to 3, so that we can undo and redo 3 steps of paintbrush operations. You need to restart FluoRender for the paintbrush history to take effect. Open the previously loaded dataset. Isolate the green channel of cells. Zoom in to find structures to select. Let's make 3 selections. The undo button is enabled after selection operations. Click undo to go back 1 step. Exactly 3 steps of undo are allowed. Click redo to go forward 1 step. We can then recover all the previous selections. Part 5. Clipping planes for selection restrictions. Clipping planes are useful when the desired structure for selection is occluded. Isolate the red channel and use clipping planes to examine the data slice by slice. Right-click one of the Z-plane controls to set the distance between 2 facing clipping planes to 1. Then, we can scroll the clipping planes and examine this channel slice by slice. Increase the output equalization setting for both red and green channels for better clarity. We would like to select the dome structure inside. Search for the first Z-slice where the structure appears. You can use the mouse wheel to quickly scroll through the slices once you click the clipping plane control. Starting from the first slice, use the selection brush to select the structure at each slice. Use keyboard shortcut Shift to enable the selection brush. The intensity signals are very low. Enable auto thresholding to find the value that works. Disable auto thresholding once the value is found. Move to the second slice once the structure is selected in the first. Click on the clipping plane control and use the arrow keys on keyboard to go through the slices. Repeat the selection for the second slice. Notice that the selection from the neighboring slice is also shown on the current slice. This is because data intensity interpolation is enabled. You have to select each slice without skipping any. However, you can adjust the distance between 2 clipping planes, so that more slices can be selected at a time. Click the XY button to set the clipping plane distance to 5. Now, 5 slices can be processed each time. Increase the brush size so that more area can be covered. When you paint on the data, only structures within the 2 clipping planes can be selected. Use keyboard shortcuts W and S to move the clipping plane slab up and down the Z-axis. Use the selection result from the neighboring slab to guide the selection of the current one. Repeat this process for all clipping plane slabs. You may set a higher clipping plane slab number to include more slices for one selection, as long as the structure can be distinguished within the slab. You can use a combination of paintbrush types for selection. For example, you may want to go back one slab to append or unselect certain structures. Once all clipping plane slabs are processed, click Reset Clips to reset the clipping planes. The selection can be viewed in 3D. We can create a new channel from the selection. Examine the dome structure without the occlusion from others. We can go to the original channel and erase the selected structure. The two channels can be viewed together with different colors. Part 6. Automatic segmentation. Separate structures can be automatically identified using FluoRender's component analyzer. Isolate the green channel and open the Component Analyzer window. Click Generate to generate components using the default settings. Processed components are selected and rendered in the secondary color. Enable 4D script in the Record/Export panel to show the components in different colors. You can filter the components by their sizes. Type in a minimum component size in the Analysis panel. Click the Clear button to clear the selection. Type All in the ID field and click Append to add all components that have a size of at least 100 voxels. This dataset has cells that can be easily distinguished by the default component generation settings. Examine the colors to see if there are any structures that have not been identified correctly. These structures seem to be two separate cells but identified as one component. Use the Component Analysis tool to confirm it. First clear the selection. Then, use the Paintbrush tool to select both cells. Click Full Component to adjust the selection to be aligned with the underlying components. Click Analyze Selection to show the component information. We can see that the selection is indeed one component. If we decide there are in fact two cells, we can manually separate them and regenerate the components. First select all components that are greater than 100 voxels. Adjust the viewing angle to find the place to separate the two. Hold down the X key to enable the Unselect brush. Adjust the brush size to a small value and paint a gap. The connection between the two cells is cut in the selection mask. Go back to the Basic panel and check Use Selection. Click Generate to regenerate the components based on selection. Wait for the calculation to finish. When we see the two cells are colored differently, we know that they are identified as two components. Component information can be acquired in the Analysis panel. Components can also be exported as channels or annotations using the tools in the Analysis panel.